The protocol described here makes it possible to identify conditions that can significantly impact bacterial persistence in a high-throughput manner. This method is readily adaptable to screen for various arrays such as drug panels, gene libraries, and more. As the method includes various sub-steps which are to be conducted simultaneously, visual demonstration can aid researchers to manage their timing to obtain precise results.
Begin by preparing microarray cell cultures. Transfer 250 microliters of exponential phase cells to 25 milliliters of fresh modified LB medium in a 50 milliliter centrifuge tube, then gently mix the cell suspension to make it homogeneous. Transfer the diluted cell suspension into a sterile 50 milliliters reservoir. Using a multi-channel pipette, transfer 150 microliters of the cell suspension to each well of a microarray plate containing various chemicals. Microarrays can also be generated manually following the method described in the text manuscript. Cover the microarray plate with a gas-permeable sealing membrane and incubate it in an orbital shaker at 37 degrees Celsius and 250 RPM for 24 hours. To make persister assay plates, prepare 25 milliliters of modified LB medium containing 5 micrograms per milliliter of ofloxacin in a 50 milliliter centrifuge tube and transfer this medium to a sterile reservoir. Transfer 190 microliters of the modified LB medium into each well of a generic flat-bottom 96 well plate. After the 24